Great. Well, we'll go ahead and get started. Welcome everyone to this month's CAIRIBU U-RIG gathering. We have some really awesome talks today. Our first talk is going to be from Dr. Jean-Phillipe Gourdine, who is assistant professor at Lewis and Clark College. Dr. Gourdine is going to be telling us about his research in host urinary glycans and something that I'm really excited to hear about. And then we have another talk scheduled from a medical student, Emmanuel Elijah, who should be joining us from UCSD. And if they don't log on soon, we might have one talk to. Oh, there you are. How's it going?
I'm good. Nice to see y'all.
Awesome. It's great to see you. So you'll come after the first talk. How's that sound? Great.
Sounds
So
good.
then, Dr. Gourdine, I'll give it over to you.
Thank you. Good morning, everybody. So I would like to thank the organizer for giving me this opportunity of presenting my work. So first of all, I'll start with some acknowledgement because we always put it at the end, but I'll start with thanking Dr. Carsten at OHSU, who was my former advisor and now mentor and collaborator now, who introduced me to the field of the urobiome, all the member of her lab and previous people who passed also in the lab. Dr. Tom Gregory at OHSU, Dr. Wolf, Tanya Sisoeva also at the University of Alabama in Huntsville, and the chemistry department at Lewis and Clark College. So this work started initially at OHSU, at Oregon Health and Science University, and we had some funding coming from the NIH, the NCATS, and then from the Circle of Giving, and then here at Lewis and Clark, I'm following on this work with the MJ Murdoch Foundation funding. So I'll start with a research context, and I know that I'm opening doors and preaching to the choir here. We know that urine is not sterile. And I'll put those questions, the when, the who, the where, the what, the why, because previous meeting, we talked about some questions we still have in the urobiome field. So let's start with the when. So when do microbes start establishing in the bladder? And feel free to add up in a comment and add up if you have some references that I may have missed, add on, please. So the earliest detection I was able to found was for a 7-month-old male, and that was published recently. And we don't know exactly when it starts, but we know that as early as 7 months old, those microbes are going to be there. Who are those urinary microbes? We know that they are really diverse. We can have viruses, fungi, bacteria, archaea, protozoan, and so on. And we know that the urobiome is really dynamic in terms of composition based on age, gender, sex, sexual intercourse frequency, ethnicity, hormone status, bladder, disease status. We can have a diversity and different types of microbes that are going to be there. For this talk, I'm going to talk mainly on the bacterial component and particularly within the female. So where are those microbes? So some people were able to show that in case of RUTI, so this recurrent urinary tract infection, those microbes were located close to the urothelium, and some maybe even inside some of those cells. And recently, Forrest and colleagues show using a urothelial microtissue modeling that some of these bugs, particularly E. coli, the commensal E. coli, can be at the cell surface, at the urothelium cell surface, and will interact with all those shedding cells. How? So how do they get to the bladder? That's still a question we don't know exactly mechanistically what's happening, but we know by comparing the genome of different bugs that are in the bladder, in the vagina, that some of them share similarity, and the vaginal microbiota have more similarity with the bladder microbiota compared to the gut microbiota. So this was published by some people who might be in the talk today and confirmed with a bigger data set here. So what what are their role in the bladder health? And what do we know about the urobiome metabolism overall? So we know that we have health-associated microbes, so lactobacilli, different type of lactobacilli for women particularly. And we know we have also diseases-associated microbiomes. So for UTI, for bladder cancer, for prostate, and for overbladder syndrome. So I'm going to talk mainly about overbladder syndrome here. And what has been shown by Lisa, Dr. Carlson, and colleague, there is an increase of bladder microbial diversity that is associated with a healthy female bladder. So when I joined the lab, Dr. Carlson lab, I was interested in the metabolism and seeing if there are some differences between those bugs in the OAB and the normal case also. So why does it matter? I know I'm preaching again to the choir. This is a CAIRIBU talk. We know that it is important for us to define what is the healthy bladder a microbe and what is the OAB and what are the metabolism, because we can either select some probiotics or some prebiotics, and we can have some inhibitor to either select or enhance the healthy the bladder associated with health. Diving on what I started to study, looking at the urine metabolome and the bladder structure to try to understand exactly how those bugs, what do they feed on, because the urine is a quasi-desertic environment. So if we look at the urine composition, different studies have shown that there are more than 200 metabolites in the urine that were identified. And among the most abundant are urea, creatinine, hippuric acid, and citrate. And if you look carefully on those most abundant metabolites, some bacteria can survive on those, but they necessitate other also compound to survive. Some other, like hippurate, are the end product of polyphenol metabolism. And some other metabolite also necessitate some other co-metabolism. So those four main urinary metabolites may not explain how those bacteria may use those molecules to generate energy here. If we look carefully at what was published, we see that amino acid and their derivative, as well as carbohydrate and their carbohydrate conjugate, are also important in the urine. And just to remind everybody, when I talk about carbohydrate, yes, we talk about glucose, we talk about the favorite food of the Cookie Monster, but we talk about more complex sugars here, indicated in this slide here, where we can have some modification of, for instance, an acetylation, we can have some fucosylation, we can have some acid that is going to be associated, and we can have more complex structure that are going to be there. Similarly, if we look carefully at the protein and glycoprotein content in the urine, we'll see that we have also a protein that is overexpressed in the urine called uromodulin, and this protein is highly glycosylated. So this is shown in this Western blot here. I don't know if you can see my arrow. Here, I'm not seeing the comment.
Yeah, we can see your arrow.
Yeah. Okay,
Yeah,
cool. Thanks. Yeah, thank you. So this is a Western blot of uromodulin here. And when we remove the sugar, we can see this shift here. So about like 30% of its mass come from a sugar, and particularly high mannose. So I'll talk about mannose in a minute, that I represent here with this green circle here. Also in the literature, what was shown recently is the fact that in the urine, we can detect unsaturated disaccharide represented here. So those unsaturated disaccharide with this double bond here are signature of microbial metabolism. So what was interesting in this paper from Han and colleague, they infer the presence of those unsaturated disaccharide coming from the gut, but they have not mentioned anything about the bladder microbe because they might not be aware that urine is not sterile here. And some people on the call, of course, Nicole Denisco and Michael, they have shown here that some bacteria can digest those glycosaminoglycan here. They have confirmation that those urinary microbe might be able to digest. And there is a correlation between some RUTI and some glycosaminoglycan that they found in the urine. So there is a lot of interest in the sugar metabolism in the urinary microbiome. And if we keep on looking more carefully at the bladder structure, we'll see here that, in fact, the bladder, exactly in this part where we have the interface with the lumen, so we have the gag layer and we have other glycoprotein that are there, such as the uroplakin, and those uroplakin have high mannose also. And on this figure here, it is the glycocalyx coming from a mouse model, but you can see all those branches represent different types of sugars. And if we compare finally with the other body sites, such as the gut microbiome, we know that a lot of gut microbes will thrive on sugars. They can be from dietary plant, for instance, or they can be from endogenous host sugars, milk, oligosaccharide, mucin, and glycans, but also from other microbes. So the question is, do microbes thrive also on those sugars and other sugars beside glycosaminoglycan? So if we look at a lot of bacteria, we know that they have a specific enzyme called polysaccharide lyase, or PL, or glycosyl hydrolases, or short for GH, that has been in a repertoire called CASI databases, where we can have a lot of information about those different enzymes and what their specificity in terms of sugar. So if you have forgotten your biochem class, so polysaccharide lyases are one type of enzyme that will cut sugars, and they will create this double bond here on their product here. And glycosyl hydrolase will use some SN2 reaction and water, and will form a different product here. So we know, as I mentioned before, that, oops, I think I'm frozen. Oh, okay. We know that sugars can contribute to the structural part for energy and can be information carrier. And we have different types of sugars. We can have glycoprotein attached with asparagine, so the N-glycans, or O-glycan with serine and threonine residue, where we can have glycosphingolipid, or we can have free like glycosaminoglycan. So do urobiome metabolize other complex sugars? So do urobiome metabolism toward host glycan differ between OAB and non-OAB group? That's those questions that are really of my interest as a trained glycobiologist who is stepping into the urobiome world. So you have different methods to get there, to trying to understand what's going on. Trying to look at the free oligosaccharide, look at the secreted protein like UMOD, or looking at membrane-associated glycans, such as uroplakin, mucin, MUC2, for instance. And we can use a biochemical approach, and we can use bioinformatic approach because we have some data also that have been shared with the community. So I'll start, first of all, with the biochemical approach and looking at the composition of free oligosaccharide in the urine between OAB and control. In a collaborative work with Dr. Song at Emory University, so we did a composition analysis of free urinary oligosaccharide, or FOS, by mass spectrometry, and trying to compare those free oligosaccharides between control group in OAB, wet, dry, and our control. So I'm just representing here one example of what we have found. We have 58 free oligosaccharides that were identified per group, and we control for age, BMI, and estrogen use. So we found that we have 6 most abundant FOS that are represented here. So on those symbols I indicated, this is fucose, galactose, and acetyl galactosamine. So this is one sugar here. So other one with salicylic acid, and other with a polyglucose here. So there was like virtually no differences between the most abundant in all those 3 cohorts, but we have like minor differences, particularly with those sugar that contains mannose here. So take-home message for this part, using the small sample size that was about 20, in OAB wet and dry. So we didn't see a huge difference between the most abundant glycan, but we saw differences between minor glycan, particularly minosylated truncated glycan. So if we put all those glycan that has mannose together, salicylic acid together, or fucose together, we saw that we have some statistical difference between groups, particularly between OAB dry and control. So the question is, are those differences and similarity due to urobiometabolism towards host glycan? So how can we answer that? So we can use the bioinformatic approach because we have some information about the bugs that are in OAB and in our control. And what we can do, we can use publicly available data that all the urobiome community have sequenced here that are available in the SRA. And we can use the CAIRIBU database that I mentioned just before. And we can use some program like dbCanPool and RCDO to mine for those genes that have a glycosidic activity and compare between our control and our case in OAB. So long story short, we use SRA genome, we use KZ, and we use dbCan. So without some caveat here, some GH and PL are well-defined and specific, while some have like a larger potential substrate. And finally, the sample case that we have used in this case were less than 50, so it was a smaller sample size. Nevertheless, we found that, that the distribution of GH and PL in the urobiome in general in OAB and as well as in the control is dominated by energy production, such as GH13, GH30. So this glycosyl hydrolase are involved in digesting glycogen and polyglucose. And we found, as I mentioned previously, some polyglucose in the urinary fossa. So this might be also explain the reason why we have those small sugar represented here. We found also some enzyme involved in peptidoglycan breaking down, but notably we found that some of the GH are involved in host and glycan digestion. And as I remind you here before, we found some small truncated N glycan also in the urine. So this could come from either the bladder cell that is shedding, but also it could come from like the digestion of those complex sugars by the urobiome bacteria. If we do a careful look of what is unique and specific and what is like statistically different, we saw there are some unique GH and PL between the control in pink and in blue, the OAB fraction, but we have some overlapping in terms of substrate utilization. What was really interesting, it was on this other case here for what was statistically significant, some of those enzyme involved in carbohydrate digestion, like PL8, found mostly in the control fraction, are involved in glycosaminoglycan digestion here. So while we have some redundancy, we found some enzymes that are found overexpressed in a control case, such as PL8 and PL12, that are involved in glycosaminoglycan digestion. And that correlates to the work that was done by Dr. Denisko's group. So I'm going to end up here with some preliminary data with what I've been doing over the summer with my student in research. So we looked at UMOD, and based on the bioinformatic data that we had, we have seen that there is this protein that these bacteria, Bifidobacterium longum, among other, that possesses this GH38, which has this manosidase activity and has been shown in other contexts to digest plant and glycan. So as I remind you, uromodulin has a lot of here N-glycan and particularly high mannose. So question is, can Bifidobacterium longum also digest UMOD here? So we have this model here where we could have in the bladder those commensal bacteria using this floating UMOD here and digest those N-glycan and go to the glycolysis pathway, ferment, and get some ATP here. So this work was done by my first undergrad student in over the summer, they did some urine purification using the ammonium bicarbonate precipitation, lectin blot with Conné to confirm that it is still glycosylated, confirm that it is really UMOD using Western blot, and some other work by culturing B. longum and with UMOD, and to see if there are some differences. So bear with me here while I'll describe what we have. So on panel A, this is the UMOD purification. So in U, this is the total urine, unbound fraction, the wash, and UMOD here was able to be purified. They did the same thing with a con A. So here we can see that those con A will bind to the high mannose content. So we have high mannose content on UMOD. And using Western blot with anti-UMOD, we know that it is a UMOD that is present here. So while waiting for my specific incubator as I'm starting my lab, I had to use a thioglycolate broth to try to mimic the anaerobic condition. And I follow up some work that was published before and use 200 microliter of MRS broth and without glucose, and I replaced the glucose by 2.5 microgram per microliter of filtered UMOD. And we have a faint here, I don't know if everybody can see, without any bacteria, we have this band here, but this band slightly disappear here. And what we see some here increase of degradation here after using different concentration of Bifidobacterial longum. So I know this is like preliminary data. We are trying to get some robust assay to confirm that part. But to conclude here, what we're able to see here first for a OAB and non-OAB group, we have seen there are some differences between the sugar that are represented in OAB and non-OAB group, but those differences are minor. But what we were able to see from the work with mining those GH and PL from what has been published with the genome, we have found significant differences between some GH and PL involved in digesting certain sugar, particularly glycosaminoglycan. So we have this confirmation using bioinformatics about the digestion of a certain sugar, and we have some suggestion also that some of those urobiome microbe can digest also some N-glycan. So I just show you some preliminary data with glycoprotein that is work in progress, and we're going to try to collaborate with people from the urobiome to get more bugs. So I know I've been in talk with Dr. Alan Wolf to get some of these bugs and to try and test what we have in the hypothesis we have generated using bioinformatics. So that's it for my talk. I thank you for your attention, and I'm welcoming any question and suggestion.
Yeah, that was a really fascinating talk. Let's go ahead and let's take a pause before the next talk and take questions for Dr. Gourdine. If you have any questions, go ahead and raise your hand or type them in the chat. I know I have a few, so I can get us started if everyone's still thinking. So I mean, your story about Bifidobacterium is fascinating. It was really refreshing to see some biochemistry in my day today, because I've been dealing with a lot of code today. So, but you know, we see Bifidobacterium quite frequently, especially longum, in the urobiomes. We have a few in our cohort patients that we think are actually dominated by that bacterium. So that's a really interesting. When you said that they could be using host glycans, are you saying they're digesting from the polymer, or they're using the disaccharide?
So for talking about the
last
GAGs,
yeah,
for the GAGs,
the N-glycans, yeah.
Oh, the N-glycans. So I think there are some of those bugs will they'll have what is called the polysaccharide utilizing loci. So they have like multiple genes like involved in cutting the N-glycans for some of them, and other bacteria might have a gene that will digest the terminal, the end part of those. Those will be able to digest like the terminated terminal fucose, and some will be able to digest mannose. So we have a complex interactive way, because those GH that are able to cut those N-glycan are not found everywhere. So it's like really selected a bacteria. So I think there is like a really interactive wall of we are studying like one bacteria at a time, but things are more complex than that.
Yeah. Are you thinking about using any microbial consortia together, multiple microbes?
That's one idea. What I'm trying to do, frankly, because I'm in a small lab, I'm in a small college where it's like teaching heavy. So I'm trying to set up like some assay where I can have student testing things within the teaching settings and collaborate with other people who might have those where it might be easier for them to test, because I've been I had some challenges like just ordering some equipment to start my lab. So I feel like if somebody is already senior and has like the technique that is working, we can like collaborate, discuss, because I don't want to be scooped and have these ideas staying in my brain and somebody else publish exactly as Dr. Denisco was mentioning about lipids.
Yeah, well, that's what we're here for, as a community, to come together and collaborate. I see that Dr. Siddiqui has her hand up.
Hi, everyone. I'm not on video today. Can you hear me?
Yes.
So JP, it's nice to see you on screen, and I'm sorry that I signed in late, so I didn't hear the very beginning of your talk, but I'm curious, I know that you're kind of talking recently about the bifido effect and that some might be cleaving, at the gag, and then in a way allowing a resource to be available to other bacteria, it sounds like. And I'm curious, we constantly see Prevotella popping up in some of our community structures. I've always wondered, because it almost seems like it's just like yet another protective thing. It may not be as protective as lacto, but like, what are they doing there? And I think Prevotella are a particular genre within an overarching bifido family. So I was curious if you've looked at that level of detail. Is it only certain bifido that you're looking at, or have you looked at things like Prevotella to see if they have these kind of similar functions?
So thanks. So I did not look at Prevotella. I'm gonna write that down. I have to look at what we have generated. So what I mentioned initially, the sample size that we had initially was a smaller one. So we use the Thomas White data initially from Dr. Wolf, what he has published, and I think it was like around 37 microbes here. And recently, there was a bigger data done that they had with like around 1,000 genomes. So we had a student over the summer in collaboration with Dr. Carsten who looked at all a bigger data set to see exactly if we have those. So I have to look in the data. I put a note for myself to see exactly where Prevotella stay. But from the mining that we have done, we can look at one particular genus and say, okay, those are those enzymes that are present. What can they digest? And I could have like more information about what are the potential substrates in regard to sugar metabolism.
Yeah, I would encourage you to do so, because it would just help to settle a question in my mind. We keep finding these, and I'm thinking like they're doing something in the environment that's facilitating, something good, but I want to know what it is that they're doing.
I'll check it out. Yeah.
Dr. Wolf has his hand up.
Yes. One thing, we also see Prevotella in lots of individuals. I'm sure that it's doing something positive. We only get it from sequencing. We tend not to be able to culture it because they're strict anaerobes. But they're gram negatives. Bifidobacteriums are gram positives. So they may be doing the same thing, something similar to Bifidobacterium, but if they are, they're doing it in a very different way. That's all.
Yeah. I think Amanda Lewis has some work in this space in Prevotella, Gardnerella, a few of these vaginal urogenital microbes in their glycobiology as well. All right, are there any more questions?
Yeah, to add to that, she's done a lot of work on sialidases.
Yeah.
Right. I mean, that's her That's her comfort zone, or sialidases. Like, for example, Gardnerella, when they co-infect Gardnerella and UPEC strain, I don't remember which one, probably UTI 89, into the mouse model. The next day, they don't see the Gardnerella, but the E. coli infection is much worse. And I know they've looked at sialidases in at least with I think a co-infection with at least one other organism, and I can't remember which one it is.
Right.
But being able to graze on the gag layer or the other glycosylated surface structures is definitely a way that bugs can survive.
Dr. Gourdine, what do we think that the absolute concentration that these are in urine? Do you have any idea?
For the digested sugar?
Disaccharides, yeah.
Yeah, so I don't have a concentration for the disaccharide. What I have,
Okay.
I have some absolute values for smaller like polysaccharide here, but those are I forget exactly in terms of a microgram per ml, but it's a low concentration.
Yeah.
Yeah.
Yeah, we usually see nanogram.
Yeah, it's a low concentration, and to a point that for some diseases that are genetic diseases like Pompe disease, you see a higher level of those polyglucose, for instance, that is like in terms of bigger micrograms for that part. But I have to double check on but it was low. It was what was like similar to what I've been found in other cases, in control cases.
Awesome.
But that could be one stuff. I don't know if anyone has looked. Oh, I see Nas as Dr. Siddiqui has hands raised.
Sorry, forgot to lower it.
Oh, okay. No worries. I was saying I don't know if anyone has looked at also genetic diseases related to a glycosylation, and to see if the urobiome composition what has changed also. That could also inform in another way what those bugs are doing if you have like elevated of those polysaccharide in the urine.
Yeah, that would be a really hard cohort to curate, wouldn't it?
Yeah, no,
I was very rare disease.
I work with, before joining like the urobiome world, I work with a undiagnosed disease network where you have patients who had an unknown disease, and the urine blood CSF were collected, and people they have that in their data bank, in the biobank. So that's one side people don't think of, but just a thought.
Yeah, sounds like an awesome resource. All right. Well, let's everyone thank Dr. Gourdine one more time. It was an awesome talk. We'll move on to our next speaker today, Emmanuel Elijah, who is a medical student from UC San Diego, working with Dr. Lindsay Burnett. And Emmanuel, do you want to share your slides? Let's see if we can get this working.
Yeah, let's see. I think Dr. Gourdine needs to, his slides are still shared, so it's not letting me.
Ah, yeah. Let's see. All right, there we go. How about now?
Let's see. Yeah. Are we able to see these slides?
Yeah, looks great.
Okay, perfect. Okay.
Cool.
All right, sounds good. Hello everyone, my name is Emmanuel. I'm a fourth-year medical student from UC San Diego School of Medicine. But actually, before I went into medical school, I actually worked for 4 years in the metabolomics lab under Peter Dorstein. And during that, that's kind of goes into my talk for today. Doing that, I really just used metabolomics as a way to try to understand what's happening within the body. And so different mechanisms, whether it was urine, whether it was CSF, or even skin, and even a lot of stool samples actually, to try to identify what the metabolomics from that space could tell us about what is happening within the body. And so for my talk today, what I kind of just want to highlight really briefly and on like an overarching theme, and just kind of understand the etiologies of the urinary metabolites on just diverse disease states. Okay. So the first question is, what is metabolomics, right? And so metabolomics on a large scale is basically just the study of small molecules. We call them just metabolites that are within cells, within biofluids, tissues, and organisms. And collectively, the small molecules and interactions within the system is known as a metabolome. And so we kind of think about metabolomics as just kind of like a direct readout of the function of the system. It's in a sense, it's like trying to eavesdrop into what the body's telling us. For in our case, in this specific talk, looking at microbes, so what are the microbes saying to each other? And the metabolites can help us infer what that is, and also it helps us kind of figure out what's happening on a phenotypic level as well. And so the way that we do metabolomics, especially in the lab that I'm in, is we first like looking at these right here. These are just the molecules that are after we go through extraction and all of that, the molecules that are found. And so we go through something called LCMS, liquid chromatography, mass spectrometry. Liquid chromatography is not the only way. Other people can do NM. Other people do gas chromatography. So it kind of depends on what it is, but liquid chromatography is one of the most common ones that is being done. And so what that does is it takes these molecules, separates them by weight, and then as it goes into the machine, it gets, whether it's processed with heat or other things, it kind of breaks down the molecules even further. And so it kind of gets this, what we call MS1, which is kind of the initial spectra. And so the initial spectra based on the weight of the molecule and things like that. And then we break that down even further to what we call MS2 data. And MS2 data is really where we get the fingerprint about what the molecule is, and helps us then go back and connect those molecules with the bigger molecule, and that allows us to kind of find similarities or dissimilarities between the molecules in the system. And then after we have the MS2, it generates this data set, the MS2 data set, and then it's then transforms into something called molecular networking. And molecular networking is a tool that we use, kind of like I mentioned before. And there's many different ways in the Dorsum lab. We do something called GNPS, which is a public forum where researchers kind of contribute their own data to help us build these libraries. And we use molecular networking to kind of see what is similar, what is not similar, what classes are they from, what classes are they not from. And as we look at the microbes, we would take the microbial metabolites, and it kind of does the same thing, which allows us to see what molecules are we finding in the sample that we've received. And as I go on later in this talk, it kind of allows us, there's some methods that we have that allows us to go back and figure out where are these molecules coming from, and what pictures are trying to paint for us. So next question is, why do we care about urine? Like I mentioned before, for me personally, I've worked a lot with CSF, I've worked a lot with stool, I've worked a lot with skin, and urine is actually something that I've just most recently started working with. And it kind of, for me personally, came across in medical school where I started to get more interest within the lower urinary tract space and the symptoms that a lot of our patients have. And for us is, again, why do we care about urine? And what we found with urine, as a lot of us know here, that one, it's going to be a less invasive way to do biomarker analysis. So we don't have to do blood draws. We don't have to do a lot more invasive things that our patients may be uncomfortable with, because number one, urine is readily available. It's something that's easily accessible. And one thing that's really unique about urine is that it actually also gives us a picture of what's going on in the body as it's filtered through kidneys. As you can imagine, different disease states have a lot of different things that are being released and different things are being excreted. Can that urine give us a picture of what's happening within the body? And that's why it's so important for us, especially after we just heard the talk with the urinary microbiome, is that the urinary metabolome connects us with the microbial ecosystem that is associated with changes in its environment. So again, it allows us to eavesdrop about the conversations that the microbes are having, and allows us to make inferences about what their purpose is or their function is within different disease states. So I just want to go across just really briefly some different studies that have shown how urinary metabolomics has actually impacted different realms. So the first is one study where it looked at pediatric obstructive sleep apnea, and looked at the oral microbiome and then the urinary metabolome, and seeing if any changes. So this was children that had snoring and suspected OSA between 3 and 11 years old. They went PSG evals, and they had urine samples from all participants at 7 a.m. every single day, and they actually collected buccal swabs. And what they found was, number one, the oral microbiome composition was definitely changed within the pediatric group, OSA group versus the normal group. We see firmicides, proteobacteria, and bacteroidetes in particular was changed. But what they also found was there was 57 metabolites that was present in the urine of pediatric patients with obstruct OSA versus controls. And those metabolites are been seen in amino acid metabolism, carbohydrate metabolism, nucleic acid metabolism, and goes on. And then further analysis that this group showed was that several of the oral microbiome changes were correlated with the urinary metabolite perturbations as well, which I thought was really, really interesting. In another study, this group looked at bicuspid aortic valve disease. And what they found was that there was multiple metabolites that help differentiate. So this plot right here just being a PCA, we're just kind of looked at the similarities between the groups. And then they did a PLSDA, which kind of tried to look at the category of the groups and to see which metabolites kind of made these changes that we are seeing in this group. And some of them were 3-hydroxybutyrate, alanine, creatine, as well as others. In this group, they looked at multiple sclerosis and NMO, and they found that there's 27 metabolites that were differentially altered in the urine from the MS and NMO patients that were associated with the ketone body synthesis or degradation, amino acid, and other things as well. And then lastly, with this group, they looked at patients with rheumatoid arthritis. And with rheumatoid arthritis, there's skeletal wasting that happens within patients who have this. And one of the questions that community has been trying to answer is, how can we assess the loss of muscle within that group? Because for one, it's invasive. Number two, it's going to be expensive. Number three, it's not going to be widely available. And so kind of what I was talking about a little bit with the urinary metabolome is that it's a less invasive way to figure out what's happening within the body. As we can imagine with skeletal wasting, some things must be excreted within the kidneys, excreted within the bodies, and may be found later on in the urine. And what they found was, yes, all of these molecules, they said were significantly differentiated that caused for the separation between the group with the low muscle mass and the group with normal muscle mass. So as we can see, even next is the sexual differences within the urinary metabolomics as well. And within the study by FAN, they literally took 60 healthy males and females, and they just ran a basic study and looked at the metabolites and based on the sex, what metabolites are increased and decreased. They found alpha-ketoglutarate, 4-hydroxybutyric acid was increased quite a bit in males compared to females. And then the fatty acids and CCI-butyrate were significantly related to the sex effect as well. So from this, we can really see just that like urinary metabolites can have the impact to show us what is happening in the body on a whole scale. And the question that a lot of us need to answer or try to figure out is, once we have those metabolites, how can we go back and actually make inferences based on that about where they come from? So for us, what is our group focused on specifically? We're focused on where the urinary metabolites originate from, and then how do they impact bladder function. We're really concerned about lower urinary tract symptoms, and we know that microbes are associated with bladder health. We know that metabolites are associated with bladder health as well, and we know that host state is associated with bladder health. Where does metabolites come from? Some come from the lumen, some come from the urinary microbiome, some come from the host, some come from other bacteria. So there's all these different things that allow us to try to figure out where these metabolites come from. So even further, we know that we've seen that some of them can be from diet and drugs. Some microbes may be from other places, some may be urinary, and then some may actually be from stool. And I will talk about the stool just in a little bit as well. And the way that we, as a group, are trying to figure out the etiology of these metabolites is through different bioinformatics tools that the Dorestein lab has developed. Something called food mass, drug mass, microbe mass. As the names imply, food mass allows us to correlate some of these metabolites to things that we see in food. Drug mass correlates metabolites that we see in drugs, and microbe mass a way to correlate metabolites that we see with the microbes. And the mass is basically it just allows for searches of public and targeted mass spectrometry data through different public libraries like GMPS, Massive, Metabolomics Workbench. And it allows us to first, based on the MS2 spectra that we have detected from our raw files, it allows us to go back and see where have these files been found. Again, this is reliant on public data about researchers and us being open for data and publishing some of the spectra that we see. And once that data is identified, we can actually figure out what data sets has this been found in. Was this an animal data set? Was this a skin data set? Was this a data set looking at microbes? And it gives us a little bit more idea of how frequently these metabolites are correlated with other things. Again, this isn't one to talk about causation. It's more so to talk about how frequently do we see this metabolites within other groups as well. And so just broad picture. So once we ran the experiment on the LCMS, we get the spectra, we put it through math and search it against public data. We have these hits from the public data, and then we actually go ahead and do a redo. And what that is you're kind of reanalyzing the data based on different metadata that the other researchers have put out. And then from there, you kind of get an association with the phenotype, the species, and the sample type. And that's the big picture of what currently the Dorsum Lab we're trying to do. And big picture, this isn't representative of our data set currently, but we have these two metabolites or these three metabolites, and we're seeing based on the yellow, like, okay, how much is this metabolite seen within these like taxonomy or these groups of microbes? And that's kind of the big picture of what we are trying to do is, again, we have these metabolites, we know that they do something, but based on going backwards, you can see where they come from, what microbes are implicated in the creation of them, and then it allows us to have a bigger picture of what's actually happening and what those microbes are doing within the disease states. So lastly, for our group and working with Dr. Burnett, where are we going? So we are currently working on assessing which factors contribute to most of the lower urinary tract symptoms and UTI pathology. We have a data set currently in conjunction with the Prevention of Lower Urinary Tract Symptoms Research Consortium. What they're doing is studying bladder health in women to identify risk and protective factors for future interventions. And the way that we do this is all of the participants, and I think we have about 400 of them, have completed a validated bladder health scales and bladder function indices. And some of the things that those scales are looking at is urinary tract infection, frequency, sensation, continence, comfort, and emptying. And so it kind of allows us to just understand the symptoms or just have the symptoms be completed by our patients. And we have 3 aims for this project. The first aim is once we have their urine analysis, we run it through mass spec, we're gonna get the urinary metabolites. First question is, based on the urinary metabolites, can we correlate them with the symptoms that they're experiencing, right? And so again, it allows us to create more questions that we can then further go ahead and answer as we go on. Second question is based on the product, can we identify where they are from? Are they from diet and drugs? Are they from the gut? Are they from different shared microbiome etiologies? Or are they from urine etiologies? And this process is going to be done by just using bioinformatics and some of the tools that the Dorsum Lab has developed. And then lastly, we want to identify whether microbes responsible for urinary metabolites are present within the gut or the urinary microbiome. No, the gut microbiome dysbiosis has been associated with lower urinary tract symptoms. So based on this, we figured that we should be able to kind of see a bigger picture of what those microbes are doing and where they're coming from within this space as well. So this is something that we're really excited about. I've been analyzing the data. Unfortunately, we don't have the prelim data for us today, but we should have some soon. So with that, I hope that we have just a brief overview of just the effects and the utilization of urinary metabolomics on different spaces. I want to thank Dr. Burnett, who's been such a great mentor. Thank Kelly Walden, who is actually an MD PhD student at UC Davis. Jasmine Zemlin, who is a staff research associate at Peter Dorstein's lab, and then Peter Dorstein as well. And with that, I'll take questions.